Welcome to Chapter 17, Part 3 of Breathing Mechanics. I've decided to include this picture right here to show you how artificial ventilation works. You typically intubate the patient and you close off any ancillary ventilation so they can't breathe through their nose, and you provide pressure and a given percentage of oxygen depending on what the patient needs in order to sustain a partial pressure which will ensure adequate oxygen saturation. Under normal circumstances, a patient's going to require 100 millimeters of mercury, but if they have compromised lung function, like in the case of a severe COVID-19 infection, that percentage of oxygen needs to be increased because of the barriers to diffusion. So you need a higher concentration and you need a stronger concentration gradient in order to ensure proper blood oxygenation and decarboxylation. In a healthy, normal person who's resting and unaware of their breathing is actually a very efficient and low energy process, requires very little energy. All you're typically doing is enlarging the pleural space by diaphragmatic contraction. So you can see right now, at rest, at the beginning of a breathing cycle, which is at the end of a normal exhalation, the diaphragm will actually be extended like this. And then what happens is that the diaphragm contracts, thus increasing the volume. You increase the three-dimensional space inside the thoracic cavity. Maybe you get a little bit of sideways motion, a little bit of intercostal slash accessory muscle contraction. But the bottom line is that you get a larger pleural space. Since there's a given amount of gas in that space, pressure drops ever so, for a split second, and then air from the outside environment, assuming there isn't any obstruction or malformation, air from the outside environment will come in and equilibrate. So it's bulk flow. During expiration, we don't usually contract any muscles. It's a relaxation of the diaphragm, and it's an elastance situation. When we're inhaling, the situation is compliance. It's how much the lungs and the chest wall allow for expansion. That's what compliance means. They comply with the command to inhale, is a way of thinking about it. On the case of expiration, the diaphragm is going to relax, and the connective tissue, the elastic connective tissue, is going to return back to its original size and shape by virtue of elastance, and thus we have a relatively passive exhalation. When it comes to inhalation, especially active inhalation, when you're going beyond normal tidal volume, we not only have muscles in motion, but we also have bones and cartilage in motion as well. So when we have an active inhalation, if we're looking at it from the side, what we see, imagine this is a rib at the end of a normal exhalation, and then we literally lift that rib up, and that changes the lateral distance from the spinal cord, and that is part of the enlargement of the volume in the pleural. Another way of looking at it is with this bucket. If we let this handle slide down, like in the case of an exhalation, let's say we're at the end of an inhalation and we relax. If you put a midline here and assume that that's the same as the spinal cord in the body, if you measured the distance from here, you would see that it is less than the distance from up here. So that's how, another way in how we can change the volume. We can contract the diaphragm in order to increase the vertical dimension of the pleural space, but we can also, with the help of especially accessory muscles up around your collarbone and or the intercostals as well, can increase the lateral dimensions of the lung. And that's why we have so much inspiratory reserve volume. We inhale 500 milliliters during normal diaphragmatic breathing, but we can inhale up to 3,500 milliliters during active inhalation. But it is a story of pressure. We change the volume with a given amount of gas inside the lungs, and that causes pressure to drop. So our alveolar pressure drops, our interpleural pressure drops, and the volume rises as the atmosphere moves in to equilibrate. So as we see pressure drop, we see volume rise. All three of these graphs, kind of like our Wiggers diagram, kind of like other figures we've seen, are all on the same time axis here. Now I want you to notice something. This is really, really, really cool. If our atmospheric pressure is so, like for instance, alveolar pressure, I want you to notice that this is all one graph. These are not two separate graphs. This is actually one graph. And so our intrapleural pressure is 1, 2, 3 millimeters of mercury lower than the alveolar pressure. That keeps the lung positively adhered to the chest wall, prevents something that is known as a pneumothorax. It's a neat little thing if you realize that these are actually all on the same y-axis as well as x-axis. This one's on a separate y-axis. Volume is rising, volume of air moved is rising as pressure is dropping. Now, if we start to increase pressure, either in the alveolus and or the pleural space, we see the volume dropping. That's because as pressure rises, the air will follow its pressure gradient out into the atmosphere. So we change pressure and then the atmosphere equilibrates. Change pressure and the atmosphere equilibrates. And you can see this again and again and again. So if 0 is atmospheric pressure, so alveolar and atmospheric are pretty similar, notice that the intrapleural pressure is actually around 3 millimeters of mercury lower. And that's very, very deliberate. It's a combination of the fluid inside here, but about 1 in every 4 to 6 or 8 breaths, we take a deeper breath. So that deeper breath, it's not a yawn. Yawning may be contributing to this, but every 4 to 8 breaths, we take one that's slightly deeper. And that's also a mechanism meant to ensure that we maintain a negative intrapleural pressure in order to keep those lungs properly inflated and adhering to the chest wall. So the combination of pleural fluid along with deep breaths every 4 to 8 cycles, the occasional yawn or so, keeps us at a negative atmospheric pressure, a subatmospheric pressure inside the intrapleural space, in between the lung and the chest wall. And so when we breathe, what we're doing is we're changing volume and the lung stays adhered to the chest wall. So like in normal tidal volume breathing, it's the diaphragm contracting and then relaxing. And then when we have maybe more active ventilation, we're using those accessory muscles and those intercostals. Again, usually when we have the exhalation, what we're doing is we're allowing the elastic recoil to bring us back. Now we do have expiratory reserve volume, and we actually have a different set of intercostal muscles that will actually force extra volume out of the lungs if needed as well. If for whatever reason we can no longer sustain a subatmospheric pressure, where either the pressure inside the pleural space is equal to the atmosphere, or in cases where pressure exceeds the atmospheric pressure, we will get a condition collectively known as pneumothorax. This is a condition where you have gas. There are other forms of increased pressure where you have additional fluid in here or blood, which can mimic a pneumothorax. Pneumothoraxes occur when the lung no longer can sustain positive contact with the chest wall and the lung begins to collapse. We can no longer alter the volume of the lung, and so we can no longer ventilate the lung properly. Like I said, it's known as a pneumothorax, and in my own experience as an EMT and paramedic, it was one of the scariest things I ever encountered. To see a man of 250, 275 pounds almost killed with just a small knife wound that you could barely see. I mean, this thing was barely a third of an inch wide. It didn't even look like a bad cut, but it was a sucking chest wound. And what was happening is as he tried to inhale, he was bringing air in this way, and his lung wasn't inflating. Unfortunately, the nature of the wound also meant that the air was getting trapped. And so as he inhaled, more and more air was coming in, and the lung was getting increasingly collapsed. A life-threatening situation for a relatively minor wound to the chest. And here's a picture. Here's a normal inflated lung here on the left. You can just kind of see the ghostly white. Look over here on the right. This is a person with a pneumothorax. You can see the lung delineation right here. I just happened to be in the emergency room at St. Vincent Hospital when an old high school friend of mine who I hadn't seen in several years just showed up, and he had this color of pallor that I hope I never see again. Well, he was experiencing a spontaneous pneumothorax. These things can and do happen because of infection. They can happen because of things like gunshot wounds or knife wounds, or in some cases, broken ribs or some sort of injury to the chest wall. A very, very serious condition that requires fairly swift action on the part of the medical team. I've already talked about these points before, but I just wanted to include these slides to just jog your memory to make sure you understand what the difference between compliance and elastance is. So compliance is the ability to stretch. So something with high compliance stretches easily. And this is a homeostatic window. You have to have just the right amount of compliance. Too little or too much leads to issues. So for instance, I actually broke a couple of ribs while I was motorcycling a little more than a year ago. Actually, these two guys up here, and the caregivers that I encountered as we figured out that I did have these broken ribs kept asking me, "Have you had any trouble breathing? Have you had any trouble breathing?" Because a flail chest, which is typically more than two ribs broken in two places, like a flail, cat of nine tails kind of thing, and it has too much compliance. You get contralateral movement. So as you inhale, the chest buckles inward and prevents inhalation. As you exhale, the chest buckles outwards and prevents deflation of the lungs. You can also see this. The next most common situation is in premature infants. They suffer from lack of proper surfactant. That's one problem. And the other problem that they typically have is when the ribs have not properly ossified. Their ribs are still primarily made of cartilage, which means that their compliance is too high, and their chest buckles a lot like a flail chest was. That's why this young person is on a positive pressure CPAP type situation. That's in order to ensure that her or his lungs are being properly inflated and deflated during this ventilation. And they're probably supplying her with surfactant as well because she's such a preemie. So compliance is stretch. Low compliance requires more force. And we call these low compliance situations restrictive lung diseases. Restrictive lung diseases are often where you have a thickening of the chest wall or the lung tissue, and so the lung tissue just won't stretch the same way. So we have what are called fibrotic lung diseases, fibrosis, the diseases that are caused by asbestosis, and so forth are known as restrictive lung diseases because you have trouble changing the volume of the lungs. This can also occur as a consequence of inadequate surfactant, either in a premature infant or even in the elderly patients. Inadequate surfactant can be an issue. Elastance, on the other hand, is the ability to return to resting volume. Classic case of low elastance is in certain forms of emphysema, where the lungs just won't stretch properly. And so what you'll see is the person suffering from the condition will actually purse their lips while they exhale, involuntarily, not realizing that what they're actually doing is trying to keep their lungs from collapsing, because the lungs stretch and then the lungs don't unstretch, and the only way they can, the only thing they can do when they go is to buckle. And so it's a low elastance situation. So a couple more slides. We're going to get to this concept of surface tension, and then I'll finish off with a slide about surfactant. The law of LaPlace can be a little bit tricky. I think this diagram's a little tricky, and don't worry about the math. We're not going to do the math. Here is the concept, and you might want to try to write this down. If you have two different radius bubbles, one large bubble and one small bubble, assuming they have identical surface tension, there has to be a higher pressure in this bubble in order for that to occur. If we flip it around, if these have identical pressures, then the surface tension in the smaller bubble is higher. So where does that go? Where does that go? Well, in physiology, we have very, very small radii in the alveolus, which means we have to have more surfactant down there. This droplet right here is many, many orders of magnitude larger than an alveolus. And so water has plenty of cohesiveness in order to form very, very large droplets inside the alveoli that would completely occlude diffusion. So we need more concentrated surfactant in those alveoli in order to break up the surface tension of water, to let water not be a barrier to diffusion. And just a quick word about surfactant. It is a mixture of proteins, lipids, and water. It does change in concentration. And you could almost think of surfactant as lining most of the pulmonary system, with more on the protein side in the upper airways and more on the phospholipid side down in the lower airways. So there is a gradient. That is because the fluids in the upper airways and conducting airway systems are really there for conditioning and trapping. Whereas in the lower airways, it's more about wetting. What I mean by wetting is breaking up surface tension. And that's why surfactant is more concentrated in the smaller alveoli, because the surface tension is highest down in there. So it's a mixture. It's a mixture, and it's secreted by the type 2 alveolar cells and other secretory cells in some of the conducting airways. One of the most common reasons for why newborns survive being born premature is the development of artificial surfactant. So two respiratory issues that preemies have is that they either lack the ossification in their chest wall for their ribs, and they have inadequate surfactant concentrations, leading to something known as ARDS, acute respiratory distress syndrome. And that happens because the lungs will collapse, and they'll also get irritated and inflamed. When you desiccate these things, when there isn't enough fluid in these lungs, or you misfold them somehow, when you get this sort of collapsing, that leads to this ARDS, acute lung injury type situation. You can see lack of surfactant in other disorders associated often with inflammation, chronic inflammation, or in some cases immune responses and that sort of a thing, where you have lack of surfactant. So it's not just a preemie thing, but it has saved lives. And if I could just make a little plug right here. Oftentimes we find ourselves interested in our specific niche. That is to say, I'm interested in things that will provide benefit in terms of whatever it is that I want to do. I want to be an occupational therapist or a physical therapist. And so I want to focus on my niche. The argument for understanding surfactant was that it was developed by an interdisciplinary team. Pulmonologists who had specialty in ventilation and respiration, neonatologists, physiologists, biochemists, biophysicists, engineers, all worked together. It was one of the great interdisciplinary achievements. And I can never stress enough, and then again, I'm plugging the whole idea of liberal arts education. The more diverse your education is, the better caregiver, no matter what kind of caregiver you're going to be, the better parent that you'll be, the better consumer that you'll be. So always keep thinking interdisciplinary. Never lose sight that any knowledge that you learn is potentially valuable. Even if it doesn't feel relevant to you at the time, stick with it. You never know when there'll be one time in your life where it will make a difference. This concludes chapter 17, and we'll be moving on to chapter 18, where we talk about gas exchange. We've got the gases moving. Now it's time to see how they move and how we get them to the tissues.